I graded the lit reviews yesterday, so Grace and I just figured out how to actually view my comments back to you in an easier way to get to the correct answers. So if you go to, there's a tab on Blackboard that says My Grades. If you click on your score for the assignment, right? Is that right? Mm-hmm. It will, or click on Lit Review 1, it will take you to what your score is, and then at that point, if you click on the score, it'll take you to what your answer was, what I wrote as a correct answer, and then my feedback to you. And you should have some sort of feedback on every single question. Okay, so take a look at that. That should be an easier way to get to this instead of having to like go back through the quiz again, or the assignment again. Yeah, I guess that's all I got to start. Did everybody see my email or announcement about homework? None this week. We're going to double up next week, but again, for one of the two, for the critical thinking, work in pairs. And actually, we are. I think we've got room actually for one group of 3. So we'll talk about that when the time approaches. The other thing to keep in mind is in a couple of weeks, not next Monday, but the one after, we're actually going to meet in the lab instead of in here. I'll make sure to announce that ahead of time again. But the reason why I bring this up is because start thinking about who you want to work with on the phage project. I'm going to send out a protocol in the next couple of days with at least the initial steps on how to perform the isolation of the bacteriophage. And part of that, you have to decide with your partner, what do you want your bacterial host strain to be? So if you want a phage that is E. coli tropic, meaning it only infects E. coli, then you want E. coli to be your host strain. Okay? I give a few examples. We have more strains than the examples that I give. But so what I'm going to ask you to do is think about it, and then also I want you to communicate with me what you want, because I need to figure out, do we have it, do we not have that particular bacterial host, and how safe do I feel with it. So like, for example, C. diff is off limits. Okay. But yeah, okay, we'll leave it at that for now. So today and Wednesday, what we're going to cover is just the general life cycle. Okay? All viruses follow these typical steps, regardless of whether it is a bacteriophage or a eukaryotic virus. There are some differences. And we will see those as we discuss specific viruses down the road. But generally, they go through 7 particular steps. And we're going to try to get through about half of those today. We'll get through the rest of them on Wednesday. Before I get started, though, what I want you to do is to take maybe 5 minutes and alone or with somebody else, put together a list. What are the 7 steps of the viral life cycle, and what's a short description for each of them? Okay? 7 steps in total. What are they? Attachments number 1. As the name would describe, what's happening here?
The virus is attaching to the host.
Okay, and if we wanted to use more specific terms, we'd say viral spike or viral receptor binding protein is binding to host receptors. Okay, that's step 1. 2, entry or penetration. The term here depends on whether we're talking about bacteriophages or human viruses. What we'll talk about is bacteriophages. The actual capsid never enters the host cell. So we call that penetration, but in eukaryotes, we often refer to this step as entry. To be honest, they're kind of synonymous terms here, so I'm not totally concerned with you accidentally using one or the other. Okay. Step 3, uncoding. This one we'll spend very little time with, but really what's happening here is we find that the capsid, maybe the viral envelope is being removed, or we start to find partial degradation of the capsid. And this is important because some viruses actually perform the next step within that partially uncoated or uncoated capsid. What is that next step? Replication and what else? Transcription and translation, all of those together. The term that the book uses and that we'll refer to describe to this whole process, biosynthesis. So we're getting replication of the genome, we're getting transcription of mRNAs, we're getting translation of proteins. After that? Yeah, I would take assembly. So what's happening here? What's actually assembling? Capsid. Genome being incorporated into that capsid as well. Next step. Yep, maturation. This one can be kind of goofy in that really what's happening here is this can occur at the same time as assembly. So we might find in the case of poliovirus, for example, we're going to see those polyproteins being cleaved, helping to form the capsid structure. That's part of the maturation process. Other viruses like HIV, they don't mature actually until after what we're calling in this process the last step, which is what? Lysis and what else? Lysis is an example of maybe this last step. We could use a more broad term. Joel? That's another example of how this occurs.
Can you say exit?
Exit is perfectly fine. Egress is another term you'll see. But both of those refer to the virus getting out of the host cell. Lysis is one mechanism by which it occurs. Exocytosis is another mechanism by which it occurs. Okay? I'm going to skip ahead a few slides real quick, but here are those major steps. So this is just an example of one type of DNA virus that's replicating in the genome, but we've got attachment, penetration, bringing it into the cell. The uncoating is the removal of the capsid shell. That DNA makes its way into the nucleus. We've got replication and transcription and translation all occurring at one time here. So I refer to this just as biosynthesis. The assembly of the virus, the maturation, and the exit as it's leaving the host cell. Okay? Every single virus follows this general theme. There are some exceptions. Sometimes the order deviates from this. Like I said, HIV, for example, actually doesn't mature until after it has exited the host cell. Benefit of this for HIV is it prevents it from reinfecting the same cell that it's just come out of. Sabrina. That's a good question. Usually it's not. It might be a place where you find, with some double-stranded DNA or double-stranded RNA viruses, you might see replication occurring within that capsid. But those RNAs that get produced actually will exit the capsid and either be that can actually detect double-stranded RNA. Double-stranded RNA in the cytoplasm of a eukaryotic cell is not normal, okay? Especially free-floating double-stranded RNA. One of the mechanisms, and I hope to get to this soon, we find that translation gets shut off in those cases. So to prevent the host cell from detecting the double-stranded RNA, some viruses will perform their replication in partially uncoated or even partially formed, in some cases, capsid shells to prevent the host from detecting that double-stranded RNA. Grace, you had a question?
Yeah, I was just curious, is integration a part of biosynthesis?
That's a great question. I think if we were talking about HIV, I suppose we could consider it part of it. I'd never thought about that before, but yeah, I don't see why not. That's a good question. Okay. Any questions on these steps here? So our goal over the next 35 minutes today and then 50 minutes on Wednesday. Cover these in a bit more detail. We won't go over every single one of them in great detail. Really, we're going to focus most of all probably on the penetration or entry step, the egress or exit step, and then we'll come back to biosynthesis as well and break apart for the 7 different classes, polymerases that are used, where these events take place, etc. I want to go back a couple of slides, and I want to show this first. So this is a plaque assay. We will see this in lab in a couple of weeks. This is a plate of bacteria. In this case, it would be E. coli. And then the E. coli is a lawn. So what you're seeing, that beigeish color, is all E. coli. Those blank spaces, that's where an individual virus has landed onto some E. coli, and they have undergone all 7 steps of replication numerous times to lead to the formation of what we call a plaque. Plaque assays are commonly used to detect viruses. It's very good to detect when the virus lyses the host cell or exits by the process of lysis. In our case, or in this case here, this can be used as a way to measure or to track the viral life cycle. We'll come back to this, but the reason why I bring this up right now is to introduce the one-step growth curve. The one-step growth curve refers to a typical round of replication in a viral host, or in a host for a virus. In this particular image, you note that the time is in minutes, so this is happening very, very quickly. This would be in culture, but they're detecting the number of plaque-forming units. We can assume each plaque represents 1 viral particle. So they're tracking the bacteriophage, in this case, by measuring phage activity or plaque-forming units per milliliter, and they're charting this over a period of time. So what they're doing is incubating E. coli, in this case, with a particular phage, and at 0 minutes, or what would that be, 3, 6, 9, 12, so on and so forth, minutes after the initial start of incubation, they take some sample, they put it into what we call soft auger, mix it, pour it onto a plate to visualize those plaques. Okay? So the plaques serve as a quantitative measurement to determine how much phage is in solution. And this is what they observe. So we've got this initial period of time where seems to be a pretty consistent amount of bacteriophage from 0 to 3 minutes. And then there's a drop. And then there's a rapid increase. Okay. These various parts correlate with those 7 steps of the viral life cycle. Attachment, entry, uncoding. We don't really have true uncoding in bacteriophages, but biosynthesis, assembly, maturation, and exit. So these correlate to that. Where is attachment occurring? Or when is attachment occurring? What's that? At 0. Yeah, really right away. So within these first 3 minutes, this is where attachment is occurring. Why do we all of a sudden, though, see a drop in the amount of phage? Where have they gone? Into the cell, exactly. This is what we call the eclipse phase. So this period of time here where we see a decrease in number is known as the eclipse phase. And during this process, what we find is that there's a decrease in the amount of infectious viral particles remaining in solution. Most of them have attached to host cells, injected their genomic material into those hosts. That's the process of penetration. And now they're undergoing biosynthesis. Somewhere between 12 to 24 minutes, we see this increase. What's happening during that period of time? That's one. What else? Probably assembly as well, maturation to an extent. Okay? And ultimately, what we find is this rapid increase in the amount of viral particles that are now in solution again as they are lysing or exploding that bacterial cell, releasing from it. And look at the numbers here. We go from somewhere around, 5 times 10 to the 8th, all the way up to 1 times 10 to the 10th number of viral particles. That's an increase of 20-fold, roughly. What this means is that there's a 20-fold increase in the amount of viral particles within 30 minutes. Bacterial cells. How do bacterial cells or human cells typically replicate? When we think of a life cycle of a bacterial cell, 1 replicates to yield us how many new cells? 2. And then 2 yield 4, and 4 yield 8, so on and so forth. There's this very consistent growth curve that we observe in bacterial cells where it doubles every so often. Bacteriophages don't play by those rules. It's this massive explosion or this one-step growth that happens in a matter of, in the case of bacteriophages, minutes. Adenovirus, hours. Day and a half or so, maybe 2 days before we observe this explosion. And if we were to track this over time, if we were to look at what happens in a culture from 48 minutes to 24 hours, we would see almost like a ladder-like step progression. In other words, where bacterial cells would grow like this, phage or viruses grow like this. In steps. Okay. And each of those steps represents, in this case, a synchronous explosion of bacterial cells or host cells releasing viral particles. Jonah.
Is that just because they all attach at the same time and then they're on like the same schedule, I guess, or is there some other factor?
No, that's what we assume. And so we can synchronize this by infecting at time 0, and usually those first couple of steps are in synchrony. After that, you see how these steps become less and less. There's a couple of reasons for that. One, they start to become a bit asynchronous. The second is the amount of host that's available, unlysed bacterial cells that are still sensitive to the phage, reduce over time, so these steps get smaller too. But one thing we can do, there are ways to normalize or to analyze this data so that we can say, hey, what's the burst size? How much phage do we get out of a single bacterial cell? So if we infected the bacteria with phage, we're starting at around 5 times 10 to the 8th. It goes up to 1 times 10 to the 10th. We said that's a 20-fold increase. What that tells us roughly from this experiment here is that from every bacterial cell that becomes infected, you get 20 phage, roughly. This isn't perfect, but we can try to do measurements like that. Using these one-step growth curves. Typically for bacteriophage, it's anywhere from 20 up to 100 will be produced from any one cell of E. coli that is infected. And that's within, we're talking minutes. All right, half hour for this event to take place. Sabrina. Yeah, so the one where I drew multiple steps, this would represent one slice of that period of time. And usually we only analyze this for one round of replication. We don't typically analyze it much outside of that. This is where we get some of that most usable information, because everything still is pretty synchronous at this point. Beyond that, we start to lose synchronicity. Okay, so I'm going to back up to this image real quick, and this is adenovirus. But just to kind of go over a couple of the terms that we just saw, so this drop again is that eclipse period. During this time, we've got biosynthesis. Typically, we think at this point the virus is not infectious. And let me rephrase that. During this eclipse period, there's been the virus has, or viral particle has attached, penetrated, and probably undergone uncoding. So at this point, we've got naked genomic material. And we think at that point that if we were to lyse those cells, we wouldn't get out, or we wouldn't be able to receive any infectious viral particles. It's not until after biosynthesis and assembly have occurred that we can detect viral particles again. What this image is showing here is if you were to measure the amount of viral particles from inside of the cell from those outside of the cell, how would this look? So in this particular experiment, what they've done is they've taken adenovirus and they have either harvested the cells and lysed them mechanically rather than allowing for the virus to do that. And you can see that we detect viral particles much earlier inside the cells than we do outside. That's because they're going through the assembly and maturation process prior to their exit from the host cell. We can do this with bacteriophage as well. We can speed up the point at which we detect them inside the host cell by mechanically disrupting those bacterial cells with chemicals like chloroform that break down the cell wall and cell membrane. There are other ways to detect viruses too. This is only detecting infectious viral particles. Okay, those that can actually infect and form plaques. One of the things we'll begin to talk about on Friday is what are those other ways in which we can analyze or determine is a cell infected with a virus or not. We can do that without having to measure plaques. Questions on the one-step growth curve? All right, let's get into the life cycle then. First step, attachment. Sometimes known as adsorption. This is what's occurring right at the beginning of that growth curve. Really, the important thing here again, recall that for a virus to be able to infect a cell, that cell has to have the receptor present that the virus can attach to. Here's an example. HIV. So HIV attaches to two receptors, the receptor CD4 on T cells and then a co-receptor known as CCR5. There are at least two known cases, there are more likely, of individuals who have mutations in both of their copies of CCR5 that render them to be resistant to HIV infection. And so what this says, or at least what this is a principle of, is saying that if an individual does not have the receptor that allows for attachment, and this could be a deletion in the receptor, it could be a mutation in the receptor, might even just be cells that don't express that receptor. If it's not present, the virus can't attach, can't infect those cells. This is why, for example, we are unable to be infected by viruses that infect yeast. We don't have the same receptors on our surface. Or for those that are homologous, there's at least enough difference in the structure that doesn't allow for those viruses to infect our cells. Second step, penetration. This is where things start to diverge a little bit. So, has anybody, when folks were kids, like really young, Anybody have those books where like it was 3 parts? It was like a head, a torso, and legs, and you can mix and match them to make different people. That's what we see with viral life cycles. Okay, we're going over the basic like different parts of it, but we can mix and match these for different viruses. Penetration is one of these steps where we start to see options being available. The options are dependent on the virus and the viral structure more than anything else. There are 2 forms of viral entry. There is fusion, and then there's some form of endocytosis. And the most common form is receptor-mediated endocytosis. Once a virus is bound to a receptor, it triggers a response to bring it into the host cell. So let's take a look at these examples. Fusion is the process in which a envelope virus, and only envelope viruses do this, have their cell envelope, after you see binding between the receptor and the viral spikes, you will find the membrane of the virus fusing with the membrane of the host cell. Sometimes this is referred to as a pH-independent mechanism, because it doesn't require any sort of pH change for this event to take place. But we can also just call it fusion. Endocytosis, on the other hand, often is associated with a requirement for a change in pH that allows for entry and uncoding at the same time. All naked viruses will enter by this mechanism, because since they're naked, by definition, they do not have an envelope, and therefore they cannot perform fusion at the cell membrane. Some envelope viruses will undergo this process as well, of receptor-mediated endocytosis. Grace?
Why does it need to be pH dependent again?
How do I want to phrase this? The change in pH will change the structure of the proteins in the capsid, or will trigger some sort of response. We'll take a look at an example in a minute with for influenza. So influenza is one that, even though it's enveloped, requires this pH change in order to trigger its entry into the cytoplasm. So I think that this may be in part review for some people, but endosomes typically around neutral pH, maybe just a little bit more acidic than that initially, but as that endosome undergoes a maturation process, it begins to acidify. Hydrogen ions are being pumped into the endosome. We might find fusion between the endosome and a lysosome, which even further drops the pH. And this pH change then is going to trigger a change in the 3-dimensional structure of those capsids, or the proteins of the virus that are found on the envelope. This can trigger, for example, uncoding. It also can trigger a fusion event between either the viral capsid, or it can trigger a fusion event between the viral capsid and the endosome at this point as well. So let's take a look at an example here. I'm going to come back to this and that. No, let's go to this. Sorry. In part, this endocytosis event is triggered by what we call clathrin. So clathrin is this. It's called a triskelion, but this it almost looks like a triangle if you look at the points on it. But clathrin is just a molecule that helps to form a cage around an endosome and helps for the invagination of the cell membrane. Oftentimes it's recruited to locations where receptors are, and it helps with this process of receptor-mediated endocytosis. One of the things we said a while ago was that viruses, in order to overcome host hurdles, they have to help restructure actin molecules, or they have to help restructure the cell the cell in order to mediate its entry into the host cell. This is one of the mechanisms by which it does it. So by recruiting clathrin to these sites of these receptors, we get this event where you get endocytosis coated with clathrin, and eventually that clathrin will kind of just deassemble from the endosome here. Okay. So a very specific example is hemagglutinin, or what's known as HA, found on influenza. HA is a protein that is, it's a trimer, a trimer of dimers. In other words, it's got 6 subunits. 3 of them are known as HA1, 3 of them are known as HA2. The HA2 here is kind of the important one for what we're talking about. So at neutral pH, HA2 is kind of folded back on itself. In other words, it's got 2 alpha helices with a loop connecting them. And it's kind of spring-loaded. At neutral pH, notice these hexagons here. These represent histidine amino acids. And I'm not asking you to know this exact mechanism. I'm just trying to point this out to give you an example of how does this pH-dependent mechanism actually work. These histidine amino acids, when the pH begins to drop, actually will have hydrogens added onto them, giving them a positive charge. Well, when that happens, they go from this locked position between these 2 loops, and they change structure so now that they rotate to the outer face. And that's as if there's an unlocking event occurring. At that point, the whole thing jackknifes open. And once it jackknifes open, you'll see right here we've got a hydrophobic domain on this protein. That embeds itself into the endosome membrane. And then slowly it pulls the endosome membrane back towards the viral membrane, which would be located on this end of HA. So that's what this event is showing here. So we get this jackknife opening, and then as it pulls the 2 back together, we start to find the viral membrane moving closer to the endosome membrane, and eventually the 2 fuse. So that's what we mean by pH dependent. The pH drop changes the structure of the amino acids in this case in some way that allows for the fusion event to occur. All right. See if there's anything else. And then the other thing, just as a reminder, all naked viruses will enter by this pH-dependent method, or by a method where they have to undergo receptor-mediated endocytosis. I'm going to pause real quick because I see confusion, so I want to make sure that we've got this straight before we, just to answer any questions and give you guys a minute to think about the Shania.
And then it pulls it back. How does that part work?
Yeah, so the way that HA is working is like it's kind of in a closed conformation to begin with. pH drops, moves the histidines, it now jackknifes open, and then slowly brings itself back together.
So like the pH starts to neutralize again?
Yeah, I think that's what's happening. I actually was trying to think about this last night, and I don't totally understand the mechanism of how it pulls itself back together. If it's, but it might be that the pH is starting to neutralize. There might be something else in here that I'm just missing that allows for this to kind of pull back together.
Does it pull itself back together after it's in the host membrane?
Yes, yes. Now this is important. Sabrina, go ahead.
So, in this picture, it's not attached to the membrane until it's below pH.
Exactly.
So the unloading.
Yeah, so like it's locked onto the viral membrane,
Right.
but you're right. The jackknife open event only occurs once pH has dropped below a certain point. So that it's kind of like it's an atomic switch in some ways, right? It's an ionic switch that allows for this opening to occur. Now, this is important not for so much the understanding how influenza does this, but there are drugs that will block the acidification of endosomes. Okay, and I think one of the review questions, we'll actually take a look at this later on. But if you can block acidification, you can block this event from taking place. Okay. So keep that in mind. Not that we use that mechanism to treat influenza, but those are the things that as we start to think about in virology, we're often thinking about, okay, prevention is important, that's the role of vaccines, but how do we actually treat? These are steps where you can start to think about treatment strategies. How do you block these events from taking place? Steps that are integral to the viral life cycle. Other questions on this? Okay. Uncoding, as the name describes, removal of maybe partial degradation of capsid proteins, creating now space in the capsid structure to allow things like nucleic acids or nucleotides to get in and out of the capsid. Also, removal of the envelope is considered a part of the uncoding process as well. This doesn't, as I kind of mentioned, some viruses will replicate will undergo biosynthesis within the capsid, so you might get what we call a partial uncoding, but not a full disintegration of that capsid. We're going to stop there. That's about all we need to talk about for this step right now. All right, so now we get into that deep part of the eclipse phase where we can't detect any infectious viral particles. At this point, uncoding has occurred, the genome has been released from the capsid, we now have biosynthesis occurring. And this, again, is referred to as the eclipse phase. We are not going to spend a lot of time with this today. I just put this up here as a reminder of, in our steps, this is the fourth one on that list of steps of viral infection. We'll come back to this and address it in more detail again on Wednesday. Okay? But in part, this should be some review, because we've already talked about this a couple of times now in our discussion of polymerases. Now, next step, assembly. These immature viral particles begin to form. Remember, the process that drives this is what? How do these capsids come together? Or how do the protomers come together to form capsimers and then ultimately to form capsids? Jonah?
Like chemical interactions with proteins.
Give me an example.
Like hydrogen bonding.
Perfect. Perfect. What is another example?
Disulfide bridge.
Not so much in this case, because those will be covalent bonds, but that definitely would contribute to the three-dimensional structure of an individual protein or protomer.
Ion bonds.
That's a good one. Somebody else, give me one more. Hydrophobic interactions as well. Great. And so remember, the key term here is self-assembly. These end up just kind of coming together at random. Once the concentration of them inside the host cell is high enough to allow for them to interact with each other. We also are getting, during this assembly process, actually somehow that genome is making its way into the capsid. This process of how the genome gets into the capsid is actually not very well understood for most viruses. We do have understanding for some, and it seems to be a scaffolding process whereby so if this is my assembling capsid, and That's a really crappy one, but nonetheless. You might have, for example, a polymerase that interacts with the inside face of the capsomers, and you find the nucleic acid being bound to the polymerase. So you get kind of a scaffolding effect, where the capsid recruits the next set of proteins, that recruits the RNA or DNA genome, so on and so forth. And again, it's all self-assembly. These occur. The assembly process occurs in these amorphous locations known as viral factories. So, does a viral factory have a specific structure? No, it's based on which virus you're looking at. Viral factories are basically sites of biosynthesis and assembly, and it can differ for each virus. Sometimes this occurs in the ER, in the endoplasmic reticulum. Sometimes it's occurring in Golgi. Other times it's just happening in the cytoplasm. So here's an example of African swine fever virus. This is an electron micrograph. The MI refers to mitochondria that are kind of lining this area around where this viral factory is located. Okay, it's not an organelle, it's not membrane bound, but it's defined by this high concentration of viral particles that are beginning to form in this region of the cell. This is a zoomed in image over here. What's the capsid structure? Icosahedral. What's going on here versus here? Maybe. What else could be happening?
Assembly?
What else? What gets assembled? Or what gets put into there during the assembly?
Genomic material?
That's probably what's happening. My guess is this is genomic material. So here's one that during the maturation process we've actually got assembly of the genome into the capsid. These ones are empty still. So which ones would be infectious viral particles? Yeah, the ones with the genomes in them. Annie?
I know you're going to go to maturation next. but
No,
what's
that's
the
fine. I think
difference-
we're going to stop here, but go ahead.
What's the difference then? Like, what's being matured? What's the difference between assembly and maturation?
Yeah, so assembly sometimes they can occur at the exact same time. There are exceptions to that, though. So maturation would be additional modifications to either capsid proteins or viral proteins, like some of the spikes on envelope viruses have to be modified with like a sugar group. That would be an example of maturation. So it's not required for the assembly per se, but it's an additional glycogen or some other polysaccharide that's put on there to give it, to modify the protein or to make it its mature form.
So the assembly is just the capsid and genomic material?
Usually that's how we classify it, yeah. It's just the coming together, it's just the self-assembly of those materials. We also can see maturation in terms of, like HIV, as I mentioned. It doesn't actually undergo maturation until it's exited the host cell. The self-assembly can still occur with some polyproteins. So HIV has one that's called GP160. It gets cleaved into GP41 and GP120, which are still associated together, but just the covalent peptide bond that's holding them together gets cleaved after they have exited the host cell. Okay, so that would be an example of a maturation step. So cleavage, protein cleavage, or protein modification, adding phosphates, adding glycogen, adding or adding a lipid moiety to the protein. All examples of maturation. All right, I'm going to stop there for today. One thing to do. So there's 4 review questions that I have for today's lecture. Probably the best one, I think, to review some of the concepts today would be the second one that's on there. It reads, compare the entry of a rhinovirus, which is naked, icosahedral, has a plus single-stranded RNA genome, and the pH-dependent entry or receptor-mediated entry of Hanten virus, which causes four-corner disease. This is an enveloped, spherical, minus or negative single-stranded RNA virus. So in other words, how does receptor-mediated endocytosis differ between a naked and between an envelope virus? Okay, that's what this one is trying to get at. We'll spend a bit more time finishing this up on Wednesday, and then anytime we have left, we'll try to do a couple of review problems. I'm not sure what we'll get with it, but we'll see what we can do. The reason why we're speeding through this a bit is because the next chapter that we look at is how do we analyze viruses? And once we know how to analyze, we can really investigate what's happening during the life cycle in greater detail. Okay? So expect that as we get into reviewing how to analyze, we'll spend more time looking at the general life cycle again. All right, have a great day, everyone.